Good. I can feel my gut relaxing.
No, absolutely. To you, my friend. So take a sip of the stout. Take a sip of the stout.
Yeah.
Tell me it's better than Guinness Neutral or Guinness Zero.
Ooh, very distinctive.
Isn't it?
Yeah.
Yeah. You can tell it's different.
Yeah, absolutely. I've got a bit of a, tad of herbalish.
Mm. That's the thing. You've got that quite bitter. There's a bitterness to add to the hops.
Yeah. Yeah.
So,
Excellent.
what do you think?
Excellent. Yeah.
Okay. So we've got one happy customer here with the Guinness.
I'll certainly use my own money
with the Gabyr stout. And to the whiskey, my friend.
Yeah, to the whiskey. Yeah.
To the whiskey. Cheers. We'll just sip.
Yeah.
So that's needs a bit more to make it like a neat whiskey.
Yeah.
But what we're going to do is we're going to recommend people put the mixer in it.
Yeah.
So what tonight I think we've got we're going to put Coca-Cola in it. Yeah. Give you a sort of whiskey and Coke. But it's not bad. I mean, considering. No. I mean, it's quite a challenge, isn't it?
Yeah. Yeah.
To do that.
I could see myself sipping on that for a while. Yeah.
Good.
Right.
Well, Jerome, lovely to see you. Thank you for coming.
Thank you for inviting me, Dave.
So you've come up from Portsmouth.
Yes, I have. Yeah.
Tell the audience, what are you?
So my main job is a university academic, but my passion is scientific research, and in particular, our nervous system.
Uh-huh.
So I've always been fascinated by human behavior. So what drives us? And the one thing that is instrumental in determining our behavior is, of course, the nervous system. So this is part of the brain.
So you're a neuroscientist?
I am a neuroscientist, yes. I'm a neuroscientist, and I do research in neuroscience, and I lecture to students. I teach students neuroscience, basic neuroscience, and I also teach them pharmacology. So historically, in the years ago, I trained as a pharmacist. So I've always been fascinated about medicine, you know, the power of medicine, you know, the value of medicine in terms of society, you know, alleviating human suffering. So qualified as a pharmacist, worked for a while, but I found it rather boring, just dispensing the medication. I wanted to invent medication. I wanted to create the medication because I'm just, you know, one thing that drives me is finding solutions. I love problems.
So what was your PhD?
My PhD was, again, so I moved from pharmacy to neurobiology. Mm-hmm. And for my PhD, which I undertook in the Netherlands, a lovely city, Groningen, in the north of the Netherlands.
Nearly Denmark.
Exactly. And I was looking at the role of a certain protein in a part of the brain that is really important for movement. So it's called the cerebellum. Yep. So whenever you learn to walk, whenever you learn to play the piano, it's this part of the brain that is basically laying down memory so that you don't have to think again, once you've learned how to walk, how do I walk? Funny thing is, I was looking at a certain protein that probably 20 years ago, they discovered that's important for stress. How do we respond to stress? So emotional stress. Yeah. But up until then, nobody had seen the cerebellum as a stress system.
Well, that is new to me as well.
Yeah. But it turns out that the highest amount of the stress protein was found in the cerebellum. So in short, I basically looked at where it's found in the cerebellum and what it does. And basically what I found is that it changes the structure of neurons. So when you apply this to these neurons, the neurons grow longer processes, which means they can form more connections, they can talk to more neurons. And that set me on a journey.
And that lays down the stress response.
Exactly. And that set me on a journey of trying to understand how do we react to stress, and how does prior stress determine our behavior later on.
And then you moved to Portsmouth and
then
set up your own
Then I moved to Portsmouth. I set up my own lab because I wanted to answer my own questions. And I continued along with the stress journey, trying to find out. And then that is when I crossed paths with GABA.
Right.
Okay.
So tell us about GABA.
So GABA is this chemical. So within your brain, if you mash it up, you're going to find a lot of chemicals. These are basically just chemicals that are produced by the neurons.
Yep.
GABA is it's a unique chemical because it's what we call a neurotransmitter. It transmits the message of the neuron. Mm-hmm. And there's different neurotransmitters. Some activate other neurons, like glutamate, and some slow down other neurons, like GABA. So GABA, I like to think of GABA as, the brakes on a car. Mm-hmm. So you're not going to get anywhere without an accelerator. Yep. So that's your glutamate, your excitatory. Right. But you'll never get there safely without your brakes. Right. So you need your GABA to be constantly modulating. And the unique thing about GABA is that it is so versatile. So it's not just a normal brake. It doesn't just shut down the nervous system. What we know is that it can function in different forms of what we call inhibition. Mm-hmm. And again, think about your car. Mm-hmm. How you use your brakes. Because sometimes you want to come to a quick stop. Yeah. Sometimes you'll be going down a steep hill and you constantly be pressing your brakes, not coming to a quick stop. And that is how we found GABA functions in the brain. And the reason it does so many things, this one chemical does so many things, is because it's targets. Right. It targets proteins that are receptive to its message. And those targets we call receptors.
And there are different targets for the different effects of GABA.
Exactly. So over the course of evolution, you've had the one neurotransmitter, but you have all these different GABA neurotransmitter receptors magnifying the function of this one chemical. So in some cells, GABA can stop a cell, and in some cells, it can just slow it down slowly. So that is the amazing thing that GABA can do.
And how did you study GABA?
Well, we can study it in a number of different ways. So I generally focus on the GABA receptors, because that's about the function of the GABA. So they are proteins. Proteins we can detect if we simply use immunology. Mm. So we can make an antibody against that protein. Yeah. We can attach that antibody to any kind of brain tissue. Mm-hmm. And if we have a little signal on your antibody, you can look down the microscope and you can see exactly where your GABA receptor is.
And you can see there's different subtypes with the different antibodies?
Absolutely. And that is really important. That's a really important question, because we now know that there are different GABA receptor subtypes in different cell types, in different brain regions. And that, well, it's important for natural function, but it's really important for pharmacology, for the medication.
So you study GABA in the brain, but I also know that you study GABA in the gut.
Yes.
Could you tell us why you do that?
Yeah. Well, as I mentioned early on, I was really interested in the stress response. Yeah. And I think anybody who gets stressed will become familiar with that knot in your stomach. Yeah. Exactly. So what we now know is that there are very tight communication pathways between the brain and the gut. Well, actually the brain and the rest of your body. So this brain-body communication.
So does the brain drive the gut? Or does the gut drive?
No. They speak to one another. So they are reciprocal connections. Okay. There's some instances when the brain would drive the gut. For example, you've taken some food, you've had some food. Yeah. You need to digest that food. There'll be signals coming from the brain to say, we need to basically propel the food. Yeah. But then there'll be some times when the gut will talk to the brain as well.
Oh, I'm full.
Yes, exactly. When you increase the amount of pressure within the gut, you basically need to send a signal to your brain, changing your behavior to say, either I need to use the loo, you need to find an appropriate place. But importantly, we know that it also affects emotions. Oh. So we know that in
gut feelings.
Yes, exactly. Feeling. But you also have these coexisting changes within the gut as well. So these GI disturbances, which are quite profound. They go beyond just motility. And we talk about inflammation of the gut. Right. So I would say they are very closely linked together. They have to function together for human.
But not many people study both. In fact, maybe you're unique in studying both, are you?
Well, no, I wouldn't say I'm unique. I think it's gaining in prominence. Yeah. I think in the past, you were a neuroscientist and you studied the brain.
Because that's what neuro means.
Exactly. You thought, well, it's the brain. We have this brain-centric view of neuroscience. The brain is most important. And yes, it is important. But we now know that what happens in the gut can directly affect or change the functioning of the brain.
And you've been studying some of our products on the gut, and also comparing them with alcohol. Tell us about that.
Absolutely. So I have this wonderful PhD student, Anna Ferreira, and in the past, I had published some work to show what GABA does in the gut and what certain GABA-like drugs do in the gut. And basically, I show that if you activate different receptors, you can either activate gut activity or decrease gut activity.
And what activity you measure?
So we're talking about contractility. So essentially motility, how quickly your intestine contracts.
So you measure how fast it's contracting.
Exactly. So there's some really important caveats to that and some important distinctions. Loads of things relax the gut.
Right.
Okay.
Not just GABA, you mean?
Not just GABA, loads of other medication as well. So, relaxation of the gut per se is not good because if it's not contracting, you're not going to get motility. No. So you're going to get constipation. So the important thing about SENTIA, the product that that Anna was looking at, is the effect was very transient.
Right.
So it relaxed. And even if you didn't remove it, the normal gut function came back again. Okay? So that means there's no long-lasting effects on motility. The other important thing was the effect was the same every time we applied it.
Oh, so it didn't wear off?
No, it didn't wear off, but also it didn't get bigger.
Right.
So I think that's really important because in certain areas of science and certain drugs, when you take one compound, the next time you take it, the effect is magnified.
Right.
And that has some kind of reinforcing effect, and you don't really want that. So if you have a good time or if you have a good effect of SENTIA, you just want to repeat that good effect of SENTIA and nothing else.
You want it reliable and consistent.
Exactly. So that's what we found. And we were able to show that it's acting via GABA.
And how did you do that?
Well, we basically have certain drugs where we can block the action of the GABA receptors.
Okay.
Yeah, we certainly block that, and then we apply SENTIA, and we can see that the effect is minimized. Okay.
So how did SENTIA compare with alcohol? You've done alcohol.
So exactly. So SENTIA actually compares with low concentration of alcohol.
Right.
So when we put in the SENTIA, you get a relaxation of the guts. When we put in alcohol, we get low concentration, so slightly less than the legal drink drive limit. Right. We get exactly the same effect. Okay. Caveat to alcohol, though, is as you increase the concentration, you get the opposite. So it actually starts stimulating, and it makes sense. If you're overindulged,
people get diarrhea when they're very drunk. So we've got a very interesting parallel then between the SENTIAs and alcohol in the gut.
Yeah.
Have you tried Gabyr yet?
Yes. I have tried it. It's quite refreshing, very pleasant, and I'm not just saying this.
But I think you've also been putting some of the contents on as well.
So we've tried Gabyr as well. So Anna has tried Gabyr, and it mimics the effect of SENTIA and low concentration alcohol. But again, its effects wear off immediately. So I think the important thing is that both SENTIA and Gabyr very closely mimic the low concentration of alcohol. But if you increase alcohol, then you start getting these negative effects. You don't get that with SENTIA because we've increased the concentration of SENTIA and Gabyr, and you simply do not get any of these propulsive effects. So you're not going to get the diarrhea, let's put it that way, with these effects. Yeah.
And you're a brain scientist. You record from neurons in the brain, which is one of the reasons we were intrigued by your previous research, and we wanted to talk with you because you're able to look at the way in which GABA works on neurons, aren't you?
Absolutely.
So explain how you do that in simple terms.
So why a neuron is a distinctive cell type, apart from other cells in the brain, is that it's electrically excitable. So basically, it has certain structures called ion channels, and they allow the movement of charged particles.
And that's an ion.
An ion, yeah. And anytime you get a charged particle moving, you get electrical activity.
So there are ions in the brain.
There are ions in the brain. So these are charged particles: sodium, potassium, chloride, that kind of thing. And anytime they move in or out of a neuron, it's the movement of the charged particle that is creating electrical activity. That is why a neuron is so distinctive from any other cell in your body.
That's why neurons fire.
Exactly. That is why they fire. That is why they're active. And that is the basis of nervous system function and human behavior. It is this coordinated neural activity.
Right.
And that happens in health. It changes in disease. So I'm really interested in seeing what happens, for example, if you're stressed, what changes. So one way we can do that is to measure these small electrical currents.
You can actually measure the current in a neuron.
You can measure the current in a neuron, and all you do is, if you have an electrode, so this is just a little glass pipette that you can put into the cell.
Into the neuron.
No. It has a little electrical wire. So it is in contact with the cell, and any electrical current that the cell passes will be caught by this electrical wire. And that is just hooked up to an amplifier, a normal amplifier. An amplifier is a device that just magnifies the size of the current.
Okay, so you can see the currents.
You can actually see the activity of the cell. So that's what we do. We can look at normal cell function. We can put a drug on, and we can see how the drug changes the activity of the cell. So we can do that in brain cells. Okay. So we can brain native brain cells. We can do that in what we call cultured cells. So these are cells that are just grown in a lab. They're not taken from a mammalian brain. And we just keep them alive in a normal incubator, and we can record as well. And then there's some other cells, again, that are cultured, and they are unique because they don't express any proteins.
Oh, right.
Okay. So, for example, oocytes, so the frog eggs.
Frog eggs. Yes. Frog eggs don't have proteins.
They don't have neurotransmitter proteins.
Right, they don't have receptors on their surface.
And they're not electrically excitable because they do not have
because they don't have those ion channels.
Exactly.
Because they're just basically dividing and turning into a frog.
Exactly. But they are unique because they're rather huge. And if you inject a protein into there, like a neurotransmitter receptor, it will start making that protein.
Right. So it has all the machinery to make the receptors.
Exactly.
It just doesn't normally do it. But what do you put into them to make them do it?
So we put in the instruction to make a protein, which is the messenger RNA.
Oh, okay.
Okay. So we put in basically the gene product.
Right.
Instead of putting the whole gene, we just put in the gene product, which is the messenger RNA, and that cell will happily start making proteins.
So you can take this cell, and you can put into it a protein that makes a GABA receptor.
Exactly.
And you can actually put in a specific RNA to make a specific subtype because there are many subtypes of GABA receptors.
Yes. Yeah, absolutely. And importantly, we can target the human form of GABA receptors.
Oh, so you put in the human mRNA?
Yeah, the human construct. So we know that.
Do you take that from humans, or is it made in the lab?
No, it's all made. It's just made in the lab.
So people know the gene, and the gene is different in the human, say, than the cow or the frog. So you get the human gene, and you make the mRNA.
Yeah, exactly.
You put it into the egg, and that egg then grows a lot of these special receptors that we're interested in.
Absolutely.
And then you can look to see whether Gabyr or SENTIA or specific ligands, specific compounds work on that particular receptor subtype.
Absolutely. And that is what another researcher in my lab has done. So
how do you actually do that? I mean, how do you actually measure the activity across these?
So this is work done by another researcher in my lab, so Rita Montero. And so she's made the mRNA. She's injected it into these cells, the oocytes. And after a few days, we know that they start making the protein. And then she used a technique called 2-electrode voltage clamp.
Okay, that sounds complicated.
Okay. So it's exactly as it sounds. So you have two, instead of just having one electrode, where I was measuring the currents, now we're measuring the voltage, which is the potential difference between a cell.
Right. Oh, inside and outside.
Inside and outside the cell. And why is that important? Because a cell is polarized. A neuron is polarized. Inside is negative, the outside is positive. So if you change the membrane potential of a cell, you will induce certain currents. Okay? And that is what's happening right now in your neurons. It's firing action potentials and certain currents are moving in and out.
Okay.
What we do is we have two electrodes. One is measuring the voltage and we clamp it, meaning we, using an amplifier, tell the cell, we're going to keep your voltage at a certain potential difference. Okay? Let's say minus 60 millivolts. We then have another electrode that is measuring the current that might be passing at that time when we're clamping it at minus 60. If you then apply a compound that opens certain ion channels, your cell is going to want to change its membrane potential. But you clamping it.
You make sure it doesn't.
You make sure it doesn't. Therefore, to do that, you're going to get a change in your current. You get a measure of your current. Exactly. So that's what we're doing. We basically clamp it at 60. And if you apply GABA to your GABA receptor, we will see this current that comes in.
So just let's go through this slowly. So you've got your cell and you've got your special, each one, you choose your receptor subtype. The cell grows it. And then you got your electrodes in and then you drop GABA on and then the receptors open and ions go into the cell and you measure the number of ions, effectively, the magnitude of the ion.
Exactly. So basically the electrode quotient of this ion movement, because we are keeping it at that holding voltage. And you can, the beauty is that it's quantitative. So if you throw more GABA on, you'll see a bigger current. Okay.
So GABA does that, but so do these things.
Exactly. So that proves, because if we did not put in the mRNA, if we just put GABA on an oocyte, you would get no current. So that tells us that your current is due to GABA, and it tells us that the cell is making your receptors. And there's no other receptors in there.
Because you've just put in the one RNA for that one.
Exactly. So now, if you put on anything else and you get a current, not GABA, anything else, you know that compound is working like GABA. It has GABA-like function.
And you've done that with Alcarelle.
That is what we've done. So we've done it with SENTIA, we've done it with Gabyr.
And with our synthetic molecules as well.
With your synthetic molecules. And we see these beautiful GABA currents, proving unequivocally.
I can say they are beautiful, actually, because the fact that they're dose-related is really elegant, isn't it?
And the important thing is that when you then pharmacologically block the receptor with your antagonist, so it's blocking it, you can actually prevent the activity. So that is the industry standard of proving that something is GABA, acting like GABA. Unequivocal proof.
And because you have put in the human receptor, then that's that little egg is a proxy for the human brain.
Exactly.
That's very impressive.
And we've gone one step further, because instead of injecting mRNA from one GABA receptor, we have taken the mRNA from a mouse intestine and we've injected that into...
Well, that'll make lots of receptors.
It'll make lots of receptors, not only GABA. It'll basically recapitulate all the proteins that are found in a mouse gut. And when we put on SENTIA and Gabyr, again, we get our GABA.
All right, so you've done it in the gut, and then you've done it in this model of the gut.
Yes, exactly.
And then you've done it in the so you've got the whole range of very specific to the gut model and then the whole gut itself.
Yeah, come full circle.
And I guess the advantage of the eggs is that animals don't die. The eggs come from
absolutely,
and you can do them forever, I suppose.
Yes, absolutely. So it's a very high throughput assay. Yeah. It's industry standard, widely used, and very reliable.
Which is what we need in order to develop new molecules or to new extracts.
Absolutely,
and new ways of putting extracts together. But you recently were the lead author on a mega review of GABA in the.
Yes.
I think you called it From Bugs to Brain. And I mean, it's 88 pages, everything you need to know about GABA from bugs to brain. Well, we've talked a lot about brain and we talked about gut. Well, tell me about the bugs.
Well, so that's, again, that's a fascinating illustration of how new science emerges, because I think if you spoke 30 years ago about talking about how bugs in your gut might affect your brain, you'd be just laughed at. But it turns out that the resident bacteria within our intestine, especially our colon, are instrumental, not only for health and well-being, but also affecting the brain as well.
And do you think GABA's involved?
Yes, because we know that some of these strains that are really important for health and well-being, they produce GABA.
Oh, they make GABA.
So they make GABA. So, and that's a huge shift because, all along we thought GABA is really only coming from neurons, from the nervous system. But now we know that it's coming from, nervous system, it's coming from bacteria. It's also coming from a number of other types of cells, like immune cells, for example. So I think there's a huge change in outlook of just thinking of this hierarchical
system. Because I started off thinking GABA was over here, and now you've told me it's in there. Now you're telling me it's in the bugs.
It's coursing throughout our body.
So when we have to start looking at whether this affects the bacteria in the gut as well as the nerves in the gut.
Absolutely. Does it rebalance or does it imbalance?
Yes. Well, I guess the technology is emerging to do that.
Absolutely. And I can imagine with, alcohol is used as antiseptic,
So probably not good. We know it reduces, it damages inflammation. I think it causes inflammation.
Absolutely. So, if the constant, long-term exposure can't be good for gut bacteria, I would imagine.
Now, one of the other interesting questions is the relationship of hormones to GABA, because I think there are some female hormones which affect the GABA system. I think maybe is it prednisolone? It's broken down to allopregnanolone as a sort of GABA.
Yes.
Do you think that could be relevant to?
Oh, absolutely. Absolutely. So we've known for a long time that GABA receptors are activated by GABA within the body, but there's other chemicals or proteins that can activate as well. And another important class are neurosteroids.
Right.
So it turns out they are really important in modulating the function of these GABA receptors. Why is that important? Because their levels fluctuate constantly. Yeah. And at certain stages, for example, after pregnancy,
the levels go very high.
They get very low. Yeah. So it's still, there's some exciting data to show how some of the system might be important, for example, in postpartum depression.
Right.
But again, we still look at it.
There's a lot to learn still.
Yes, exactly. Yeah.
So this GABA receptor, just say a little bit more. Yeah. It sounds very complicated. Yeah. What does it actually look like?
Well, if imagine... So the first thing is that it's not one protein.
All right.
It's a complex. It's a complex of proteins. So it's a complex of proteins we call subunits. Mm-hmm. So the cell will make the subunits.
Yeah.
And in an amazing feat of organization, the cell will assemble these subunits into a complex, 5 of them, and they come together and they form an ion channel. So remember earlier I spoke about ion channels.
Yeah.
It's basically a portal where ions can move
So they make a tube. The proteins come together, make a tube.
Exactly.
And the ions go through the tube across the cell membrane.
It's almost like a gate. So it's a portal tube where ions can move in and out of the cell.
Right.
And as they move, that is when you generate the electrical activity.
And then on this 5 proteins together, you've got binding sites for various things.
Exactly. So you have, I won't go into too much of the details, but there's in the human genome, we talk about 18 subunits. And so we have alpha, beta, gamma. Generally, you have 2 alpha, 2 beta, and some type of gamma.
Right.
And they, in terms of how they are arranged, so generally you have an alpha, beta, and a gamma. And on the interface between the alpha and the beta, that is where your GABA molecule binds.
Right.
So GABA is released from its neighboring cell, and that's where it binds to on that cell. But there's other parts of this receptor that are susceptible to be activated by other compounds.
Maybe like alcohol.
Like alcohol. So alcohol can bind to the GABA site depending on the amount.
Yeah.
But there's another site called its allosteric modulatory site.
Right.
Basically, if something is bound to that site and GABA happens to be bound, the effect of GABA is magnified.
Right.
So it stays a bit longer.
It enhances.
It enhances the effect of GABA.
Are there other GABA receptors?
Yeah, so there's generally 2 classes,
Right.
GABA-A, GABA-B.
Right.
So GABA-A is the ion channel. That is the 5 protein subunits that come together to form the complex. It's in the cell membrane, and it's this tube, the portal from the inside to the outside.
Right.
There's another class, and that activates very quickly. So it's just ion movings. So the signal is in milliseconds, fraction of a second.
Right.
But you also have a very slower form of GABA, where if GABA binds to a GABA-B receptor, its effect takes a bit longer.
Right.
So we're talking maybe minutes, maybe seconds, multiple seconds, and minutes.
Right. And that's a very different kind of receptor.
That's a completely different type of receptor because when it binds, it relies on a cascade of events before GABA's full effect takes place.
And that cascade occurs inside the cell?
It occurs inside the cell. But either way, it's slowing down the activity of the cell.
So when will you make your sort of model of your human receptor in your egg?
Yeah.
You put in different subunits to come together to make this. It's not just one protein, it's actually 3 proteins.
Exactly. So we can put in an alpha, a certain type of alpha, a certain type of beta, and a certain type of gamma 2. And that's important because we know that different receptor subtypes do different things in the brain, but pharmacologically, they also have different effects as well.
And this little egg knows what to do because once it's got the RNA, it just does what the RNA does.
Absolutely. We can replicate what everybody else has done. So we can replicate all the pharmacological effects of compounds that are currently available. And then when we use the bespoke synthetic compounds, we can actually see that it's doing the same thing.
And we're very interested in using this technology to see if we could discover new molecules in plants that would do this. Is that possible?
Oh, absolutely. Absolutely. Because once you've got the receptors, all you need to do is find a way to extract your target compound. Okay.
And then how do you get it onto the receptor?
You simply apply it. So you dissolve it in solution, and your egg is sitting there. You have your 2 electrodes, and you're simply flowing solution over it. Okay. You're simply bathing it in the solution. And as soon as you put it on, within less than a minute, the channel's open.
So lovely technology.
Yeah. Instantaneous. If you look at certain conditions like Parkinson's, so these are human conditions, Parkinson's, Alzheimer's, and you assay, so you look at the population of bacteria in the intestine, in the colon, completely different. The big problem facing the industry, the science at the moment, is that it's all correlative. So you have a medical condition and you see this change in the bacteria.
It comes first.
Exactly. So you don't really know whether the changes in bacterial population are causing the condition or the condition is doing the thing. But there's clear evidence that what happens microbially in your gut has a profound effect on the brain.
I was speaking the other day to a very famous ex-rugby player called Rory Lamont. He used to play for Scotland. A terrible injury. He played about 28 times for Scotland. A terrible injury, and became very depressed because he was chronically ill with his fractured leg, and he got infected. And then he said, "I was struggling," and then they put me on antibiotics, and everything crashed. And it sounded like the antibodies were killing the bugs that were keeping him functioning. And then he became catastrophically depressed as a result. Does that make sense?
Oh, man, absolutely. I mean, it's very difficult, for the one case to say unequivocally, but, scientifically it makes sense because we know that there's certain strains of bacteria that directly correlate with health.